Hi everybody, it's your AP Biology teacher, Mr. Poser. Today we're getting into topic 6.3, and that is on transcription and RNA processing. So yes, we are getting into the gene expression stuff today, finally, and we're going to be talking about the actual function of what DNA does and what DNA is actually used for. In our last topic, we talked about how it gets copied and how it can get passed down from one generation to the next because of, cells' ability to copy their DNA. But what is it actually used for? Well, it's to make proteins, right? And proteins, their job is super dependent on their shape, and therefore their shape is super dependent on their amino acids. And therefore, their amino acids is super dependent on the code that cells read in order to put the right amino acids in the right place. And that's what DNA is about. That's what gene expression is. So this is the language of life, everybody. Anything, any living thing has DNA, and it's able to use that DNA to make a proper protein, provided it has the right cellular machinery and the right enzymes with which to do that, right? So this is the language of life. A's, C's, T's, and G's in a very, very complicated sequences are used to build any protein that exists, all right? Which is kind of amazing, right? Some proteins are like, what a gene is, I think is like, their average gene is like 20,000 bases long or something like that. So it's pretty amazing that this is all exists, and it's super complicated, mind-bogglingly complicated. So we're going to go over the basics of kind of some of the first steps of this gene expression today. All right, but let's get a general overview here. What is DNA used for? It's a code for making proteins that all cells can read. Okay, and the sequence of A's and T's and C's and G's carries information that cells can transfer from one place to another in order to build the right proteins. And proteins, like I said, do everything, right? So a cell cannot function without its proteins being in the right shape, and therefore its DNA, its genetic code needs to be correct as well. All right, but now here's the thing about that, right? DNA is used to make proteins, but there's a molecule that goes in between that, right? And it kind of serves as an intermediate, right? So it's kind of like if DNA, all the DNA inside the nucleus is a library, right? It's kind of like checking out a book from the library a little bit, right? So like if you have a school project, way back before the internet, right? Okay, I was in high school when the internet existed, right? This is not me personally, all right? I'm not that old. But back in the day, right, if you had a school project, something you could do is go to the library and check out a book to help you with your school project, your paper, your essay, whatever, right? Now, do you need every single book in the entire library? No. Okay, you're just going to take that one book and maybe you might copy some notes from it and then use it on your project or your essay, right? That's kind of how gene expression works a little bit. All right, and if that process of checking out the book and maybe copying some stuff from it, that's what's called transcription. All right, and this is the process of making RNA, this intermediate molecule, is called transcription here. So in this video, the first part of it is all about RNA. It's the intermediate between DNA and proteins, the library and the final product, right? In other words. Okay, so we're going to talk about a couple different kinds of RNA here in just a second, but I want to go over the basics just in case we need this. Maybe you talked about this in a previous biology class, or you already know this from like 7th grade or whatever. But let's talk about the central dogma of molecular biology. All right, and this is the rules kind of that all living things follow. All right, all living things have DNA. All right, unless you're like a virus, but we don't know if those are living things in the first place. Anyway, living things have DNA, right? And that DNA is used to make RNA, okay? DNA does not become RNA, all right? But RNA base is a little transcript, a little piece of it is built based off of a little segment of DNA called a gene, all right? And that's what's called transcription. That's what we're going to get to today, all right? But the road doesn't end there, right? RNA is then translated. The code that's copied from DNA is translated into the amino acids that will eventually build a protein. And proteins do everything, right? Proteins have so many different functions. Okay, we've talked about quite a number of proteins already in this class, and we're going to talk about more. But yeah, transcription and translation, these two twin processes here allow living things to use DNA to make a temporary copy called RNA, and then that RNA is translated in order to make the right amino acids for the right proteins. And all living things follow these rules, so that's why it's called the central dogma. All right, another schematic here. Here's DNA. It's in the nucleus, okay? Let's just say right off the bat here that not all cells have nuclei, right? We're talking about eukaryotes here. Not to say that this does not happen in prokaryotes, because it does. It's universal here, all right? But just the schematic is in eukaryotic cells here. All right, so DNA is stored in eukaryotic cells in the nucleus. All right, and then this process of transcription occurs where it takes a little piece of DNA called a gene and makes a temporary copy, what we call pre-mRNA from it. M stands for messenger, and you'll see why that is in just a second. Okay. And then the second part of this video is going to be about what's called RNA processing, right? So this RNA, this pre-mRNA is modified a little bit so that it can be brought out into the cytosol, okay? It can be taken out of the nucleus. It can be modified just a little bit so that it can be brought out to the cytosol. And this is not pre-mRNA. This is now called mRNA, or it's not this is what's called the primary transcript, and this is what's called mature mRNA, but I'll get there in a second. All right. And then once it's matured a bit and it's modified and it's ready to be taken out of the nucleus into the cytosol, it's going to get transported to a ribosome. And if you remember ribosomes from way back in the day, they're the protein factories, right? What they're going to do is this process called translation. They're going to read that mRNA 3 letters at a time, like AUG, for example, and make a corrected sequence of amino acids with that. And those amino acid chains eventually become proteins, all right? Now, that's looking a little bit into the future, but I want us to get a big picture view of what we're talking about here this unit before we get into some of the technical stuff, especially when it comes to translation, okay? And how gene expression is regulated as well. That gets pretty complicated, okay? But this is your 30,000-foot view, as some people say, all right? This is generally how it works. All right, I put up here the sequence of bases, the A's and C's and G's and U's in RNA determines its structure and function. Hey, that sounds familiar, right? The sequence of amino acids in DNA, in a protein determines its structure and function. The sequence of A's and C's and G's and U's, the nucleotides and RNA determines its structure and function as well. And we're going to talk about 3 different kinds of RNA. All right, the first of which we already kind of took a look at. It's called mRNA. That M stands for messenger RNA because it carries the message from the nucleus to the ribosome. It's an intermediate, right? It carries a code transcript from DNA to the ribosome. And here it is again, right? Here's this RNA molecule being made based off of this DNA. It gets processed and then shipped out of the nucleus so that it can go to the ribosome. This is mRNA. It contains the DNA code, a temporary copy of that DNA code. And we're going to look at that a little bit more detail in the next topic, right? But there's also 2 other kinds of RNA that are going to play a role here in gene expression as well. The first of which is called tRNA. T stands for transfer RNA. And we're going to look at this again a little bit more in 6.4, right? But this tRNA, this is this blue molecule over here, as indicated by this blue arrow, that's going to match up with 3 base pairs in the mRNA, which are called codons, and bring over an amino acid to a ribosome so that the ribosome can kind of piece it together and start to make a protein, do some dehydration synthesis and form some peptide bonds between those amino acids in order to make a protein. Okay, so transfer RNA, we're going to see more of what that does in the next topic. And then rRNA, R stands for ribosomal RNA. Ribosomes are made of ribosomal RNA. All right, it's the building block of a ribosome. All right, so there are 3 different types of RNA that you're going to have to know. mRNA contains the code that's of that gene that's brought over from the nucleus to the ribosome. tRNA are RNA molecules that are going to bind to mRNA in 3 bases and bring over an amino acid, and rRNA is what makes up ribosomes. All right, so those are what you got to look for. Okay, and as I said before, transcription is the process by which RNA polymerases, hey, that sounds familiar, it's like DNA polymerase, right? Except we're making RNA now instead of DNA. Use a segment of a single strand of DNA, which is also called a gene, to form a new RNA molecule, right? Whether that's mRNA, tRNA, or rRNA. But we're going to be mostly focused on what mRNA does. For the rest of this video here. Transcription, this is what we are on, and you can see it happening right here. Check it out. Here's my good old DNA. It gets split apart. Hey, that sounds familiar too. RNA polymerase comes in, slides through, and produces a new RNA molecule, a single-stranded RNA molecule that's based off of this template strand over here in that DNA. All right, and that's what's called a gene, right? This little section of DNA. Oh, this looks familiar too. Yeah, I used a similar animation as what I did in the last one because transcription is a little similar to replication in a way. All right, it's similar in that, these two DNA strands get separated, 3' 5' 5' 3'. All right, and then a new enzyme, not DNA polymerase this time, RNA polymerase, is going to bind and slide along in the 5' to 3' direction and produce an mRNA molecule. Or what we call a primary transcript, this initial copy of this gene. All right, and once again, a gene is a segment of DNA that codes for a protein. All right, so there's my RNA, and it's going to get shipped off. It's not going to stay in the nucleus. It's got to get sent to a ribosome, right? It's going to get shipped off to a ribosome. It's got to exit the nucleus so that it can be translated and used to make a protein. All right. Now, here's my primary RNA transcript. I left it pink here. Maybe the pink was the same color as the newly synthesized strand of DNA from last video, but now it's RNA. All right, just bear with me. Pretend it's RNA now, okay? The pink line is. All right, RNA polymerase produces the mRNA transcript, or what's called the pre-mRNA, which is then modified before it leaves the nucleus, right? So we looked at before how mRNA gets copied, or it gets transcribed, right? And then exits the nucleus and goes to the ribosome. But some stuff has to happen to it first. All right, before that's done, it has to be modified or processed. And that's the second part of the topic, or the title of this topic. It's transcription and RNA processing. This is what processing is going to look like. For a pre-mRNA, something gets added to the 5' end called a GTP cap. All right, GTP, hey, we brought that up before. GTP, guanosine triphosphate. It's kind of like ATP. ATP is jaded cousins because it's not as popular, right? So GTP gets added to the 5' end, all right, before it exits the nucleus. And something else gets added too. A whole bunch of A nucleotides get added to the 3' end, or that's what's known as the poly A tail on the 3' end. All right, so before this RNA can leave the nucleus, these two things need to be added to it. And the reason for that is that the GTP cap helps with ribosomal recognition. Basically, without the GTP cap, the mRNA is not going to basically kind of bind with a ribosome so that it can start to be translated. And the poly A tail stabilizes the mRNA molecule, makes sure it doesn't get degraded by, say, like a lysosome or something like that. And it says, hey, I'm supposed to be here. I'm supposed to go to a ribosome. All right, so it stabilizes that mRNA molecule, prevents it from being broken down. Okay, so those two things are added to the mRNA transcript before it leaves the nucleus. And then there's one other thing that happens too, and that's called splicing. Okay, this pre-mRNA is spliced. Unneeded sections of mRNA are removed and necessary sections are put together. So think about like if you've ever edited a video before. I know I have, right? You can take out parts that you don't want and delete them, and then put the parts together that you do want and stick them together so that it forms one cohesive. Unneeded sections of mRNA are removed. Those are what are called introns, and I put those in red up here, right? So these unneeded sections called introns get taken out, all right? And now the blue sections, which are called exons, those are necessary sections that the cell wants to keep, are stuck together, kind of like editing a video, right? And this RNA is ready to go, presuming it has its 5' GTP cap and its 3' poly A tail. It's ready to go. It's ready to get shipped out of the nucleus and sent to the ribosome. Okay, in other words, introns are excised, which means they're cut out, and exons are retained, which means they're stuck back together. All right, now here's what can make this a little interesting here. All right, you could have the same pre-mRNA, but depending on how it's spliced, you could get two different proteins. How is that? Well, that's due to alternative splicing. You see what I did with the colors up there, right? I just switched up the colors, right? So like different sections of the same mRNA transcript or pre-mRNA can be spliced out and result in different mature mRNAs, right? So this one's different from that one, right? I switched around the colors, right? So depending on what's an intron and what's an exon, you could end up with a different mRNA than what you had previously. All right, but depending on what's an intron and what's an exon, what's getting removed and what's getting kept in. All right, and a different mRNA results in a different protein, right? So one gene can result in several different proteins, all right? Maybe that's, that's some of the genetic stuff that we talked about in the last unit. It has to do with alternative splicing. Okay, so there you go, all right? Alternative splicing, you can trigger different translations based on what's an intron and what's an exon. Okay, so as a recap here, I'm going to run through everything one more time just in case you missed anything or I missed anything. RNA is the intermediate molecule in gene expression. It allows for protein synthesis based on the code in DNA. All right, DNA is the ultimate language of life. RNA are the temporary copies that are used to, carry information from the nucleus to the ribosome. In eukaryotes. All right. mRNA carries transcript code from DNA in the nucleus to the ribosome. That's M stands for messenger because it's actually going from one place to another, carrying the genetic message. tRNA transfers. T stands for transfers amino acids to a ribosome based on the mRNA sequence code. We're going to look at that more in 6.4. And rRNA, R stands for ribosomal, makes up ribosomes. And the process by which RNA is made is called transcription. RNA polymerases produce new RNA molecules based off a DNA template strand, kind of similar to how replication works. All right, last couple things here. RNA polymerases only produce RNA in the 5' to 3' direction, very much also like DNA polymerase. But instead of, going in two directions, we're just making one strand of RNA, so we don't really have to worry about all that stuff. But before the mRNA is sent to the ribosome to start making that protein, it has to be modified before it leaves the nucleus. And of course, this is just in eukaryotes, right? Because, prokaryotes don't have a nucleus. And gene expression happens simultaneously. Transcription and translation happen at the same time in prokaryotes, but we'll talk about that later too. The poly A tail and the GTP cap provide stability and ribosomal recognition for that modified mRNA, so it can be sent to the right place and is not broken down. Introns from a pre-mRNA are cut out and removed or excised, while exons are stitched together, and that's what's called splicing. And depending on what's an intron and what's an exon, you could get different mRNA transcripts, and that's what's called alternative splicing, and that might come back later on as well in this unit. All right, let me know if you have any questions, and we'll see you next time.